Hi, everyone, and thank you for joining us today. I'm Arslan Arif, the publisher and founder of Endpoints News. I'm honored to be your webinar moderator today. Today's panel is sponsored by Agilex, and our topic is non-clinical and clinical pathways for rapid vaccine development in Australia. And today we're joined by Dr. Kurt Sales, the knowledgeable director of immunoassay at Agilex Biolabs. Also, Dr. Peter Tapley, the director of toxicology at TetraQ, an Agilex Biolabs company, and also Dr. Drew Brockman, the head of toxicology at TetraQ Agilex Biolabs. So if you have any questions during today's presentation, feel free to submit them using the Q&A button at the bottom of your screen. We look forward to seeing what you have to say on today's topic. And from here, I'm going to hand it off to Kurt to start today's discussion. Kurt?
Thanks very much, Arslan. Hello viewers, and welcome back to this webinar series hosted by Agilex Biolabs. In today's webinar, we're going to discuss briefly the non-clinical and clinical pathways for rapid vaccine development in Australia. And by way of introduction, I'm going to briefly focus on an overview of the Australian landscape, provide a snapshot of the Australian advantage, Agilex Biolabs, and what we do, before launching into today's topic, hosted by my colleagues, Drs. Peter Tapley and Drew Brockman from our TetraQ toxicology service. And they will provide an overview of our toxicology service as well as the requirements for non-clinical vaccine development before launching into a little case study in order to provide a little bit more information. I'll then provide some information on the bioanalytical requirements and general assays used for vaccine studies in preclinical to clinical before we wrap up today's webinar. So really why many people choose Australia is the rapid setup and advancement from preclinical to clinical. For example, a full CMC package isn't required for getting into the clinic. A really comprehensive investigative brochure outlining all of the preclinical data, pharmacology, safety, efficacy data is all really that's required in order for the ethics committee to review and make a recommendation to move forward. And this means that we can get into the clinic in as little as 6 weeks after the ethics committee has approved the trial. And so this fast to clinic is really common in Australia and really enables the clinical trial to progress in a really rapid manner. In terms of expertise, we have key opinion leaders, and our expertise in Australia is second to none. Most of our key opinion leaders have also trained overseas with extensive connections and extensive connections with international colleagues. Most importantly is that the data that is generated from all the clinical trials within Australia is internationally recognized and compliant with all of the regulatory agencies, including the FDA, the EMA, the MFDS, TFDA, and the RCH. What's really lucrative for small biotech companies that's in the burning rather than earning phase, earning less than $20 million revenue per year, is the really attractive R&D cash rebate set forth by the Australian government. And this really is a 43.5% rebate on all of the activities associated with the trial. And this could be from the manufacturing right through to the preclinical to the clinical trial conduct and wrap up. So it really is a very lucrative R&D tax rebate, which is bringing many investigators from abroad into the Australian environment for rapid setup. In terms of COVID-19, we've been relatively fortunate over the last 15 months, with minimal impact. It's been business as usual with all of the biolaboratories, CROs and phase 1 units staying open throughout. For the bioanalytical component, we can provide fast startup once materials have been procured for both small and large molecule bioanalysis and allowing real-time analysis of data to really enable that advancement of the clinical trial. So what is Agilex Biolabs? Well, we're a leading bioanalytical laboratory in Australia. We've been around for over 2 decades. And we work globally across all countries with a footprint in Adelaide for our bioanalytical laboratory and in Brisbane for our toxicology laboratory. So our main service lines, as highlighted in the center panel, is preclinical toxicology, and then bioanalysis using the streams of tandem mass spectrometry for small molecule, and classic ligand binding assay for immunobiology and bioanalytical immunoassay PK analysis. And then overarchingly, over both platforms, we can provide biomarker analysis, either using a multiplex or singleplex analysis, depending on the type of biomarker required. We're an ISO accredited company, accredited to ISO 17025, and importantly, we're also OECD GLP accredited. And this means that we can do GLP tox studies and run clinical studies to clinical GLP. So what do? We provide an end-to-end solution for regulated bioanalysis, as I mentioned, from preclinical toxicology, which focuses on early stage discovery toxicology and pharmacokinetic studies, GLP-compliant toxicology studies to support clinical development, and then PK assays across all species, including humans. And this enables us to either do de novo assay development or method transfer, and validation to both EMA and FDA bioanalytical guidances. We have a range of instrument platforms and a range of capabilities to be able to do high throughput. We worked on all molecules from novel chemical entities to novel biological entities, biosimilars, antibody drug conjugates, and so on, in order to be able to measure free and total PK measurements. For those biologics, we can provide a tiered approach to immunogenicity, from screening confirmatory to titer, and then neutralizing antibody assays. And then we can provide a variety of additional pharmacodynamic measurements, either looking at target engagement for those immunological targets, or other ligand binding assays in order to determine mode of action of drug, or biomarker assays for secretion of soluble molecules into matrix. For immunology assays, we have the state-of-the-art BD-FAC Symphony A3 flow cytometer. It's a 5 laser 20 color instrument, which really enables us to do multi-parametric analysis. And I'll touch on some of that a little bit later on. We also have Luminex and MSD and Lispot or Fluorospot, specific for vaccine studies. And more recently, we've expanded to gene therapy areas in order to provide a more therapeutic approach for RNA vaccines that are coming through to the market by installing a digital droplet quantitative RT-PCR platform. So I'm now going to hand over to my colleagues, Dr. Peter Tapley and Drew Brockman, who will take us through some of the non-clinical pathways for rapid vaccine development in Australia.
Okay, thank you, Kev, and hi, everybody. So, yeah, I'd like to start off with a quick overview of TetraQ and Agilex Biolabs currently. So our service scope, as Kurt has mentioned, is exploratory and GLP rodent toxicology services. We can offer second species studies via partner labs overseas. So our facility has had 16 years of continuous operation in a purpose-built rodent toxicology facility located in Brisbane, Australia. We have an experienced team of study directors and really high-quality animal techs to conduct our studies, and supported by QA also. And we operate to OECD Good Laboratory Practice standard for our GLP for our tox studies. As Kate has mentioned, the R&D tax rebate scheme is applicable to non-clinical studies, so a vaccine study, GLP vaccine study, for example, is eligible for that tax rebate system in Australia. We've been able to operate throughout the COVID-19 pandemic and continue to do so, pretty much business as usual. In the rodent field, we have no issues with animal supply, so typically we can start studies within 4 to 6 weeks of contract approval on their local animal ethics submission. Next slide. So just to drill down a bit further into what we do, so we're really focused on high quality toxicology services supporting vaccine, small molecule and biological drug development. Those studies range from early exploratory studies, single repeated dose acute studies in rat and mouse to support early phase drug development, also pharmacokinetic studies, more for the small molecules, and immune response studies, again, might support vaccine development. The major part of our business is the GLP toxicology studies, and these correspond to rodent studies conducted to GLP compliance in our facility, ranging from acute single-dose extended toxicity studies, for example, up to 13 weeks duration, typically. And then second species studies conducted by our partner labs. So a major application of our studies is supporting first-in-human studies in Australia under the Clinical Trials Notification Scheme. So we really represent an experienced provider also of, in particular, GLP vaccine non-clinical safety studies to support first-in-human in Australia. And we, as Kurt's mentioned, we have very strong bioanalytical support for our vaccine studies provided by the Kurt's immunoassay and immunobiology group, and also small molecule capabilities from the LCMS team at Agilex. Next slide. So just talk a little bit about specifically about our about vaccines and non-clinical development of vaccines. So first of all, it's important to understand that there's a number of regulatory guidances which are applicable to vaccine development, and I've listed some of these here: EMA and WHO guidelines for vaccines, and they provide a good framework for design of toxicology studies. And the ultimate objective is to then provide evidence of safety prior to the first clinical trial. Go to the next slide. Thank you. So vaccine study design it's relatively straightforward, but there's some certainly key considerations for a successful study design. First of all, similar to all first-in-human enabling tox, the toxicity study test item should be comparable to the clinical test item. So typically it would be a engineering run from the GMP facility manufacturing the clinical test item, or the clinical test item itself. It should, so the derivation of that test item should allow you to make a claim that it's comparable to the clinical test item. Typically, vaccine development requires a single species for tox rather than two species. So rodent, for example, is generally an acceptable species. Rabbit is also used on occasion. Toxicity study design needs to be informed by the clinical design. So the dose route should match the clinical dose route. Dose frequency should equal or exceed the frequency of administration in the clinical setting. So if there's 2 doses of vaccine to be administered clinically, then typically there would be 3 doses in the non-clinical study. The dose interval should be similar to the clinical dose interval, but it can be reduced, so you can do an accelerated design, especially if you've got evidence that you have you can induce the intended immune response with a shorter protocol. So a 3-week or 4-week study in humans might be condensed to 2 to 3 weeks in a rat study, for example. The dose, typically there's a requirement to administer the human dose or a maximum feasible dose. So depending on the dose route, there may be animal welfare limitations which would not allow you to administer the human dose. For adjuvanted vaccines, there's consideration to providing to including an adjuvant-only group, or even an antigen-only group is the other alternative. And one of the key considerations is also to make sure that there's good assessment of local tolerance at the administration site. So parameters to look at during a in-life study, then after you've organised your design to match the guidances and requirements of your clinical study, fairly straightforward, typical tox observations, clinical observations, including the injection site for that local tolerance. Body weight, temperature is often measured, looking for increases in temperature after a treatment, for example, and looking for that to go back to baseline. Terminal examinations are really standard GLP tox, gross and microscopic pathology, clinical pathology. One important endpoint is a serum antibody titer to measure the immune response using a validated immunoassay method. So that goes back to making sure that you've picked the relevant species. So it's important to demonstrate that in the safety study that you have a appropriate response against the target. So that's, just a quick overview of the sort of considerations for clinical and non-clinical design then for a vaccine. And next slide, I will hand over to Drew Brockman to talk in some more detail about a case study.
Okay, thank you, Peter. A case study and what Service Agilex Biolabs can provide is a preclinical safety assessment performed in support of the University of Queensland molecular clamp SARS-CoV-2 vaccine. So the objective of this study was to assess the toxicity of the S-clamp vaccine when administered by 3 doses separated by 2 weeks to spray dolly rats. So the table here shows that the design of the study entailed 3 groups. There was a saline control, which acted as a comparator, an S-clamp antigen-only group, and then the vaccine group, which comprised the antigen mixed with the adjuvant MF59C.1. The rats were treated on days 1, 15, and 29. A subgroup of 10 male and 10 female rats in the main allocation was terminated on day 30 to 31, while a second recovery subgroup of 5 male and 5 female rats were terminated on day 43 after a 2-week recovery period. The purpose of this recovery period is to assess the reversibility of any findings detected in the main study allocation. So the design is recommended by the WHO guidelines, where 3 doses are given to support a clinical regimen of 2 doses, and the interval between the doses can be reduced in the preclinical tox study, 2 weeks in this instance to support the clinical design 4 weeks. Next slide. In this preclinical toxicity study, 3 treatments of the vaccine administered at the max feasible dose via the intramuscular route were well tolerated. Findings of mild mixed cell inflammatory reactions were detected at the injection site, which showed evidence for reversibility and were not considered adverse. The vaccination also elicited a strong immune response to the vaccine antigen in the rat model, which is an important readout for the preclinical study. So overall, the study was critical to the development of the UQS clamp vaccine and supported the progression to clinical trials in Q3 of 2020. The preclinical results were published in Clinical and Translational Immunology, and the clinical results in Lancet Infectious Diseases. And with that, I'll hand back over to Kurt.
Thanks, Drew. So I'll briefly touch on the clinical landscape. Thanks, Drew. And so with that, I'll briefly touch on the clinical landscape and bridging the transition from preclinical to first in man. So the clinical ecosystem comprises of a variety of different stakeholders and partners, and I've listed a couple of these on the slide, from laboratories to CROs to hospitals to phase 1 units and others. As I've mentioned in the first couple of slides, and Peter and Drew would have mentioned as well, is that the ethics committee is the sole point of decision making for moving the clinical trial forward. And so what's required for launching into the clinical phase is a complete investigator brochure, which encompasses all of the preclinical safety data, the pharmacology data, the toxicology data, and that together with all of the other supportive in vitro data running up to that, forms a package which is evaluated by the ethics committee, and the ethics committee would then look at all of that information to provide a recommendation as to whether that it's likely that the program is safe enough to go into humans. So once the ethics committee has actually made that decision and given the approval, the clinical trial can then commence. And startup is then facilitated by means of the clinical CRO, and I've listed a couple of the major CROs in this slide. And they work together with the hospitals and the phase 1 units in order to set the trial up. Partner labs, either conducting the safety laboratory tests or the specific bioanalytical laboratory tests, form part of the add-on services to provide a safety package for that clinical study. So I want to talk a little bit about the bioanalytical considerations for vaccines trials and just highlight some of the general assays that are generally considered for these types of trials. So the most common assays and platforms used for vaccines are competitive ligand binding or quantitative ligand binding assays, either an ELISA-based assay or an MSD or mesoscale type assay. And these are sandwich assays which are really geared towards picking out that antibody that is generated against the vaccine in the human or animal species. And so it is almost a type of quantitative PK assessment of that warranted immunogenicity reaction and a titer in order to determine the relative levels in EC50. In order to determine whether the memory T cells actually have or can respond, we can use Lispot and flow cytometry in order to do recall response reactions, and I'll provide a couple of slides on these in the next few slides. And then you can also provide some neutralization assays or plaque reduction neutralization tests, where you can incubate your serum with cells, plate them out under agar, and look at plaques and inability of the virus to infect the cells as a method of determining neutralization or neutralizing antibody effect of those antibodies present within the serum. And then finally, one can do hemagglutination assays also to detect and quantify antibodies to virus. So I really want to focus on the main two that are most common, and to focus on a quantitative ligand binding ELISA-based assay and what this actually looks like. And so an ELISA-based assay is a sandwich immunogenic assay where you really are plating out the antigen, which is generally the vaccine target on a plate. And that vaccine target then is going to bind to any antibodies generated within the animal or the human against that target. So for example, if you are immunizing the animal with a spike protein, the animal is going to generate antibodies against that spike protein, and that is what you're wanting to see. So this assay is then geared towards pulling out those anti-spike protein antibodies and then being able to quantify the relative amounts. And so I just put an example from the Watterson paper that Drew had mentioned in his presentation from the SCLAMP assay and vaccine study, just to provide a little bit of a snapshot of what data actually looks like in terms of the ELISA EC50, which is quantified by means of this type of sandwich assay that I've put on this slide. So it's a really powerful measurement of the immune response generated against the vaccine antigen, and quite well quantitative. In order to determine whether the memory cells have actually can elicit a response and can remember the response towards that immunogenic antigen, we can use Lispot. And so the way that we do this is get cells in. Cells are plated and cultured under various conditions. And then we can stimulate these cells with an immunic antigen to induce a recall response. In this example, we're stimulating PBMCs with CEFTA, which is a pool of MH2 restricted viral peptides. These selectively activate CD4 T cells. And then I'm also using more general stimulant for CD3, CD28, which stimulates all T cells. And we can then detect specific cytokines that are churned out following stimulation of these cells using a plate-based Lispot, either as a single plex, or we can use a multiplex fluorospot to target several cytokines. And by doing this, we can actually extrapolate single cell producing cytokines. And then we can, by means of magnetic bead sorting, we can actually pull out specific cell populations and delve even more deeply into lineage. So what does the data typically look like? So in this slide, in the bottom, for cells, we have cells in media alone. And then in the top, we have cells that are cultured at different concentrations of either the CEFTA pool of peptides or the CD3, CD28. And so what happens is that the plate has antibodies against interferon gamma plated out, and you plate your cells on top of it. And if the cells then produce interferon gamma, it then binds to that interferon gamma antibody on the plate and forms a spot. And so all of these little black dots are spots that are interferon gamma proteins secreted from these cells. And so what's evident from this slide is that you get a differential response if you stimulate T cells only, CD4 T cells only, compared with if you stimulate all of the immune cells. And so one can really use this tool as a powerful mechanism to look at how individual cells actually can behave once they've actually seen an immunogenic peptide or protein, and how their functionality has changed over the course of the trial. We can become a little bit more sophisticated, and we can use a multi-spot approach where we can use 3 different cytokines that are spotted out onto a plate as an antibody, and use the same approach for stimulating cells with either a pool of MH2 peptides or CD3, CD28. And this is even more powerful because what it does is it enables you to identify those cells that either only produce interferon gamma or TNF alpha or IL-2, for example, and then those cells that actually produce all of those peptides. And then by means of magnetic bead sorting, we can actually tear down those lineages of cells as well in order to identify specific cells and cells that produce a specific response in the presence of a certain peptide. By means of flow cytometry and magnetic beads sorting, we can actually pull out those different lineages. So magnetic activated cell sorting is a very powerful tool, coupled with multiparametric analysis from a flow cytometer, really enables us to be able to look at a quantitative analysis of these different cells that are producing different cytokines and switching on different cell surface proteins. And so in this technique, we have antibody that's bound to nanosized beads, and the beads are added directly to whole blood. They then pull out a specific population of cells. We can stain those with specific antibodies, and we can then put them through a flow cytometer and actually do multiparametric analysis. And this is what it basically looks like, where you can gate on forward and side scatter on singlets, gate out the doublets, and gate out all of your lymphocytes, and then using those antibodies, you can provide a really sophisticated tiered structure where you can have a look at the populations of cells that are either different lineages that you've pulled out of those magnetic beads, and then you can identify specific proteins, either intracellular or extracellular proteins that are differentially regulated in those specific cell populations in response to a peptide. It's a very powerful tool and used quite commonly for a variety of different immunological profiling response reactions, but also for vaccine studies as well. So in conclusion, sort of non-clinical evaluation of vaccine safety requires consideration of the guidelines, regulatory guidelines, and the intended clinical use of the vaccine, so clinical trial preclinical trial design is very important and really needs to marry up very closely with the clinical trial design. TetraQ and Agilex Biolabs company is a very experienced provider and very well designed studies, robust studies of non-clinical safety studies in rodents to support vaccine development. And we've provided a recent example and publications that are freely and widely available by means of PubMed. And our immunoassay and immunobiology teams can then provide that ongoing support for the analysis services for both the non-clinical and clinical vaccine development. So I hope you found some of this information interesting and useful for being able to move vaccine study from preclinical through to clinic, and considerations for different trial design and considerations in general for being able to start such a study and move it forward to first in human. Thank you.
All right, Kurt. Thanks so much. Let's move to our Q&A session now for the panel. So I've got a couple questions here. And my first is this: what are the main considerations to get started with the preclinical portion of the vaccine study?
Yeah, thanks, Arslan. I can address that. And we talked a little bit about that in the presentation. But yeah, really critical is having your test item in an appropriate form to conduct the study. So you need material which is very similar, comparable to the clinical material. So from the same facility, and if it might be an engineering run prior to the production of the GMP clinical material, or it might be the clinical material. If it's an adjuvanted vaccine, then you need appropriate supply of the adjuvant as well, so that you can take both into the non-clinical study. One other consideration is ensuring you've got the right species. So at some point, there should be a study undertaken, and TetraQ can do this, immunize the target species with the vaccine and generate some immune serum from that species to demonstrate the vaccine's active in that species, and also to generate immune serum, which can be used to development of an immunoassay to measure antibody titer. But yeah, really test items probably the critical rate limiting step in our experience with sponsors, having that available and ready to go. Thank you.
Okay, very, very good. Okay, another question. So are multiple species needed to provide enough data to go into human clinical trials?
I think Drew might address that.
Yeah, generally only a single species is needed for vaccines to be taken into clinical trials here in Australia. For other non-vaccine test items, there may be a requirement for multiple species, but for the vaccination pathway, a rodent, a mouse, a rat, or as Peter mentioned, rabbits are also commonly used as a single species.
Okay, all right, very good. And so for the bioanalysis, what critical reagents are required to build assays?
So thanks, Arsalan. So yeah, the test item is very, very important, as that's the main reference item that's used. But then also an antibody generated against that test item is very important to use as a surrogate to be able to set a cut point. And so, for preclinical, this could either be an immunized animal and pre-immune and post-immune serum. And for the clinical study, we prefer to have something that's a little bit more purified. So it really depends on the nature of the vaccine. There are various different formats nowadays, from mRNA vaccines to protein vaccines. Most vaccines, delivered within a viral vector epitope, and so consideration is to what the vaccine antigen is. So, for example, for COVID, if it's a spike protein, and you're generating an antibody against that spike protein, in order to capture that antibody, you need the spike protein to plate out on the cells. So it's been able to get those reagents that are specific for the actual study, and so that would encompass the immunogenic peptide or protein and a surrogate antibody, which would act as something to be able to use to evaluate those antibodies generated within the animal or human again. So those are the main critical reagents that we would require for this type of study.
Okay. Oh, well, Dr. Kurt Sales, it is always such a pleasure to hear your perspectives on this stuff. And Dr. Peter Tapley and Dr. Drew Brockman, glad to have you here too. And with that, we're going to have to conclude today's webinar presentation. And a big thank you to our sponsor, Agilex Biolabs, and again, our panelists for sharing their expertise with us today. If you would like to rewatch this webinar or share it with colleagues, a link for on-demand viewing will be sent tomorrow. I'm Arslan Arif for Endpoints News. Thank you for joining us, and we hope to see you at a future webinar. Thanks, everyone.
Thank you.